one or
Nope, this is everyone, and Robert is going to record this, so if you do have to leave, we can just send you a copy of what happened after.
Oh, it's okay. Don't. I won't have time to like review a recording. But if there's salient information, send me a note, or we can also have another conversation.
Yeah, I mean, I'm recording it to send to Murray as well.
Oh,
I don't know if you
that would be great. That would be great. So, okay, since we're all here, I just wanted to thank all of you three for really keeping this project going, and Robert also for keeping it going during the COVID. I mean, it's not been easy to work, I'm sure, for anybody, and it's hard to sort of keep a collaboration going even when everything is normal. So it's really great. Sound like there might be some promising results, and Peter's work is at the phase where we're ready to publish. So maybe we could publish this stuff together. So I think that would be ideal. So anyway, I'll stop talking.
Yeah, if we only have 30 minutes, I think I'll just
since I only have the 30 minutes, and I'll listen. And
let me just share my screen here. Can everyone see my screen?
I can see it.
Okay, all right, let me jump right in here. All right, yep, so as you all know, I've been doing some structural modeling of Peter's fluorescent mistranslation reporter, specifically the proline to serine reporter. And I'm gonna do a little bit of background like I did last time on Rosetta, just to brush up here. And we know that Rosetta can do de novo folding, some docking, full on like protein design for specific tasks, even DNA, RNA stuff. And it's been developed by many developers from all around the world. And it was the first software to successfully predict de novo folding from sequence alone. So it's a very powerful software. And there are lots of protocols like for specific applications that are like pre-written, and there's tons of documentation on this stuff. Like for example, here we have specific antibody protocols, some docking protocols, peptide protein docking, AB initio. So, and there's many more here than I've included. So before we understand how Rosetta works, I just want to go over how a more traditional molecular dynamics simulation would run. So here we have the starting conformation of a protein in its energy landscape, and we have the final conformation. And this squiggly line just represents the folding trajectory. So the goal of a molecular dynamics run would be to follow the folding trajectory as it would happen in real space. So we're concerned with all the relevant transition states, all the local minimum, and in using force fields and things like that, we try and follow this curve as closely as possible. Now, the downside of this is that it's very computationally expensive, and the simulation trajectory is subject to energetic barriers. So in this case, let's say we have this large energetic barrier right here. Sorry, can you guys see when I move my mouse on my screen? Yeah, you can.
I can.
Let me just, maybe if I switch it to laser pointer here. So if we have, let's say, large energetic barriers like this, in some cases, molecular dynamics might not be able to find the proper transition state, and, well, the trajectory might not get to the proper final conformation. Rosetta, on the other hand, doesn't look at the folding trajectory at all. It uses a Monte Carlo-based approach where we're looking at random parts of the potential energy landscape of the protein. And so it just looks, Rosetta just looks for local minima of structure conformations. And if the energy scoring functions are correct, then we should be able to identify the lowest energy states and hopefully the final conformations that we're looking for. Now, the advantage, or the way it does this, is that Rosetta trajectories are dictated by empirical data rather than force fields. So it's all dictated by structures that are already in the PDB. And this method is orders of magnitude less computationally expensive than molecular dynamics. But for this particular project, why I thought Rosetta was good for this project is that it's the Rosetta algorithm is not subject to energetic barriers. So let's say we have a large folding barrier like here before. It doesn't really matter because Rosetta doesn't look at the folding trajectory at all. We're just looking at final conformations. And in the case of designing or modeling a mistranslation reporter, I thought this was appropriate in this case. So the way Rosetta samples conformations is first we have some global conformational sampling where you have fairly large perturbations in the protein backbone. And then followed by local side chain sampling where side chains are packed into their more, just packed into proper orientations. And both of these sampling steps are empirically guided. So it's all based on structures that exist in the PDB to sample only the most likely conformations, because there's like an infinite way that you can fold these proteins. So that's how Rosetta achieves sampling, but just as important as sampling, you have scoring, which is calculating how likely each of these structures are. So in molecular mechanics, like I mentioned, you have force fields that dictate the simulation trajectory. And you have things like the Lennard-Jones potential, you have Coulombic laws, and like a lot of different angle torsions and strains that are all calculated at each time step of the simulation. And this, like I said, is computationally very expensive, and the computation time scales exponentially with each individual atom, with each additional atom. So the larger the protein, your computation time will increase exponentially. Rosetta, on the other hand, scores its structures also empirically. So it does sampling empirically and it does scoring empirically. So what does that mean? Here we have a histogram of all chi angles within the PDB. And as you can see, like there's areas, that are of higher density, right, of chi angles that are adopted in the PDB. And it's through probability statistics like this that Rosetta uses to calculate how likely a structure is. And so what is its relative energy? And you can stack up a lot of these terms, one on top of the other, to get a more accurate scoring function. And like people have devoted their entire careers to expanding the energy or the Rosetta scoring function. Now, the advantage to this over molecular mechanics is that the computation time scales linearly with each additional atom. So you can simulate much larger structures than you would with molecular mechanics. And in a lot of cases, much more accurately. So I gave this talk yesterday to a few people from our lab. So I just gave a little bit of a rundown on what you guys are trying to do here. So I'm not gonna go through this, but basically we're just trying, what Peter has done is he's designed a serine to proline mistranslation reporter, and we're trying to model it to understand the mechanism of dysfunction. Why is this mutant non-functional? And in doing so, perhaps we can identify a way of designing other mistranslation reporter mutants in silico. So we're all familiar with, well, just the overall structure of these fluorescent proteins. So mCherry, you've got a beta barrel structure, and then a helical central peptide running through the middle, in which you have the fluorescent chromophore. And it's secluded from solvent. It's just buried way inside the protein. And so I did a first initial run of just this. Sorry, I should mention this is a crystal structure of mCherry, PDB code 2H5Q. And so I took this crystal structure and just ran a Rosetta minimization on it just to see what would happen. And so this is just, well, trajectory of one Rosetta run. And so you can see it just goes through perturbations in the backbone, and it samples some local side chain conformations. And finally, we get this output structure here.
Hey, Robert, can you comment real fast about solvent? Is there any solvent? Is it implicit solvent? Does it not matter because it's really based on this knowledge-based potential where there is solvent in the crystal structures that it's based on? Anyway, let me know what about that.
Yeah, so I think somewhere in the scoring terms, there is like some solvent accessibility terms and like what's exposed into solvent and what isn't. It's all somewhere in the scoring terms, but for the most part, I think it's just based on empirical structure data. So like how do we know that these, yeah, just like side chain packing and rotamer conformations. But I believe there is somewhere in the scoring function a term for solvation energy. I don't know the exact specifics, but it seems to do a pretty good job. But anyways, like how do we know that this actually represents the protein in real life, this output structure? Well, we use statistical analysis here. So if we take this output structure and we plot its RMSD to some reference, in this case, it's just a starting structure, and we calculate a Rosetta score for it. If we repeat this, hundreds or thousands of times, the distribution we would expect is kind of a converging cone, or we call this a funnel, right? Where you see all the structures converging on a Rosetta score at a particular RMSD. So this would be indicative of a successful Rosetta run. And the reason why we get this kind of distribution and why you don't get the same structure every time is because Rosetta proceeds through a number of random functions throughout its algorithm. So every time you run Rosetta, you're going to get a different output. And so in this case, every time you run anything in Rosetta, it's a good idea to run it many, many, many times and look at the distribution that you have, right? Because just one structure doesn't tell you anything, right? So we have this funnel plot, which is what we call it, and if you see this sort of distribution, then it's a very high probability that the structure at the tip of your funnel represents the most likely conformation of protein in real life. So using this pipeline, we would generate like our mCherry mutants of interest, generate 1,000 minimized structures, and then calculate Rosetta and energy to generate a funnel plot. And using the funnel plot as a kind of readout of whether or not a mutant is functional, we would expect a functional mutant to look like this and a non-functional perhaps converging at a different RMSD. And initially I thought that this would be what we would be using as our readout for whether or not a mutant is functional. But unfortunately, when I did this with just straight wild type, we had the signature of a non-functional mutant coming out. And I dug into this for a while and tried to troubleshoot some things. And as it turns out, the reason why I was getting a significant number of errors was because of the way Rosetta was treating the chromophore. Right, so the chromophore itself, it's there's not enough data in the PDB of like how the different rotamer conformations of this chromophore and how it behaves, and so the output wasn't very accurate because there's not too much empirical data for this chromophore. So Rosetta didn't really know what to do with it. So I went and took a look, and it seems like another group who was modeling GFP deletion mutants ran into the same issue. And the way they got around it is rather than simulating the mature chromophore, they replaced it with the what's it called, with the precursor peptide, right? So for GFP, you have serine, tyrosine, glycine. And in the case of mCherry, we have methionine, tyrosine, glycine.
So this seems like it's much better from everything you told us, right? Because there's no data, there's little data in the PDB for such chromophores, which only happen in GFPs anyway.
Yeah, exactly.
But there's a lot of data for those other three residues because they're normal. So
Yep.
this is really cool. Okay.
Yep. And so although like the structure of this precursor isn't really the same as this chromophore, we can make, oh my goodness. We can make some, or what they did is they made some functional inferences based on like what this final structure looked like. And I'll get into how they did that later. So this was their published pipeline. Basically, they did the same relaxation that we just did, but with, at this step, they remodeled the chromophore to the precursor. And from there, they added in their deletion mutants of interest and did some more minimizations to get an output structure. And then with their output structures, they used some pretty clever machine learning methods to decide whether or not the models were functional or not, right? Because there is so much. There's actually like databases out there of just GFP deletion mutants and like real data of whether or not they're functional. And because there's a lot of data out there, they were able to generate machine learning algorithms to decide whether or not these structures are functional. And from that, they can then predict other GFP mutants. Now, unfortunately, there aren't any databases like this for mCherry. There's actually not too much mCherry mutational data out there. Certainly not enough to generate a machine learning algorithm. So I had to come up with a different way of predicting the functional status of each mutant. So I'll get to that at the end. But first, let me go into my kind of adapted computational pipeline here. So step one is to delete the chromophore of mCherry and replace it with the precursor, methionine, tyrosine, glycine. And then there's a pretty recent Rosetta protocol called the kinematic loop closure protocol, or otherwise known as KLC. And this uses, I'm not sure if you're familiar with Rosetta fragments, but it uses small 3-mer and 9-mer fragments from the PDB to rebuild a structure. And so I'm using this protocol to rebuild the chromophore precursor as well as a lot of flanking residues. So residues 56 to 74. And the way I've picked 56 to 74 is it's because it's that central helix that runs down the center of the beta barrel. So I figured that would be an appropriate amount to remodel. From there, we'd identify the highest fidelity models, add our mutations of interest, and minimize the entire structure using Rosetta Relax. Right, and from there, we could do our analysis to analyze any structural and energetic differences and see if we could come up with a mechanism for mutant dysfunction. So let's start here with this step with the KLC loop model step. So this is our starting model that I used, and I say starting model, but really the position of this red loop, which is being remodeled, it doesn't matter at all. So what I'm trying to say is the starting model has absolutely no bias on what the final conformation is going to look like. So Rosetta is rebuilding this red loop completely from sequence alone. So this is what just one single run of Rosetta loop model looks like for KLC. So as you can see, there's like an insane amount of perturbation that happens. So there's very little bias that goes into building this model. The only bias really being the rest of the protein, right? Because it's being treated as a static structure at this stage. Right, so this is what it's building, and this is our output model here. And as you can see, the simulated precursor maps very well to the actual crystal structure with the mature chromophore. So Rosetta did a pretty good job at modeling the central loop. And so, like I said before, just one output structure doesn't really mean anything. We have to do this 1,000 times. So in this particular case, I think I did it 960 times. And we look at the distribution. And we're actually seeing a funnel distribution come up, with the exception of this kind of cluster over here. And I'll show you what that looked like before. So here's an overlay of every single one of these 900 structures, right? And so you can see a lot of these are forming peptides here in the center as expected. Some of them are doing these, some of these models are doing this ridiculous thing where it's building this peptide on the outside of the barrel, which again speaks to like the lack of bias that Rosetta uses to build these models. And so these like structures here on the outside correspond to this little cluster of structures over here. But if we take a look at the lowest energy structure, this is what it looks like. It goes through the center. We see a nice helical peptide going through the center, and it maps even more closely to the mCherry crystal structure. Okay, so this indicates like pretty high confidence that this is what the precursor looks like, what the precursor peptide looks like. So this is the model that we're using going forwards, right? So we identified the highest fidelity model, and now we can add a mutation, the S146P mutation, and minimize the whole structure using Rosetta Relax. So one more time, the Rosetta Relax program does structure perturbations on the whole backbone, not just what we simulated before. So this is what we got for wild type distribution like this. And I also tested A145G as a functional control, since I know you guys have data for this particular mutant. And of course our S146P non-functional mutant. So if we look at the distribution of these plots, the spread in RMSD and in score is actually very, very tight comparative to other Rosetta runs. So this kind of indicates that pretty much all of these output structures are almost as equally likely, right? So there's fairly little spread between each of these models. We're just zoomed in really close on RMSD and score.
Yeah, you don't have a big spread, and I mean your RMSDs aren't that high, right? I mean they're all pretty close to the crystal structure. If, you know, 0.7, it's not right, but it looks to my eyes. And if you wanted, you could do like a principal component analysis to really demonstrate this.
So actually, I tried that.
But it just looked to my eyes like the S146P is shifted up and to the right.
Exactly. It is shifted up and to the right a little bit.
What we're looking for is where does the ensemble of structures shift to, right? And so that's kind of would be the signature of something being destabilized, lower scores, right? And higher RMSDs, basically.
So that's exactly it. Like when we're dealing with a spread that's this tight, we can't just look at a single output model. We have to look at the kind of distribution of all output models, and that's where all the information lies, not just in a single structure. And you're right to have noticed instantly that we have a kind of shift of up and to the right. Although this is such a small shift in score that it's probably just due to a small kink in the backbone from the proline. I wouldn't, you know, I would think this would be a pretty small window readout if we were to be testing the functionality of mutants this way. So I decided to dig a little bit deeper here, right? And like, how else can we analyze the structural and energetic differences here to identify a mechanism. Well, let's go over like all the possible mechanisms of why this mutant is dysfunctional. First, maybe we have like a global fold disruption, right? Maybe the protein just completely isn't folding properly. Not as likely, but could be possible. Another possibility is the local environment surrounding the chromophore is disrupted, right? So you have in these fluorescent proteins, you have an extensive hydrogen bond network, which places the chromophore in the correct orientation and shields it from solvent. And the local environment is very important for the functionality of the chromophore. So perhaps that is being disrupted. And finally, maybe the mutant doesn't work because the precursor itself is unable to mature into a fluorescent chromophore. And so these are the kind of three mechanisms that I decided to explore here. So first, if we overlay every single one of these structures, and I analyzed mean differences of torsion angles between each structure. So this gives us a measure, like just a very easy view of backbone flexibility, right, in what areas. And between all three mutants, we see pretty much an identical flexibility profile. I don't see any significant differences between the three, with the exception being at the actual position where the mutation occurs. There's some backbone differences there, but other than that, I don't see any other global differences, which suggests to me that the mutant isn't folding improperly. Perhaps maybe it's energetically slightly less stable, but I don't think that's enough to say that it's not folding properly. So we'll discount that for now. And second, let's look at maybe the local environment is being disrupted. And so in all these models, I took a look at all these residues that were published and is known to be important for chromophore stability, right? All these residues that are involved in this extensive hydrogen bond network. And again, I didn't see any differences between wild type A145G and S146 mCherry. Like if this chromophore were to mature, the stabilizing hydrogen bond network wouldn't be affected post-maturation.
But Robert, can you just go back? So isn't that S146 there that's on the screen?
S1.
That's S146 hydrogen bonding
Yeah.
with the tyrosine here.
Yep. So that's
if that's a
so
protein, I mean, that's eliminated that hydrogen bond. So that should change at least somewhat, and maybe it's the maturation.
Yep. So yeah, you bring up a good point there.
Yeah, we should look locally at what happens, or almost like, yeah, I don't know. Just a quick suggestion, like maybe just make the mutant and then see what happens to the conformation of the pre-chromophore, so to speak.
Exactly. So that's where I'm getting to.
Oh, you're getting there. So sorry. I got to leave in just a minute.
Sir. So I thought I can
recording.
Oh, sorry. If you got to go, we can.
Since I'm going to go, I'm going to listen for like another 30 seconds and then I'll go off. Let me get in just some quick suggestions. For future work, and maybe you already thought about this, we should probably crystallize the 146 mutant because your work makes it look like might be worth doing. And Peter showed it's soluble, fine soluble, just like the wild type. It's just not fluorescent. So it should be possible to crystallize it. And or another thing to think about would be folding experiments. Based on what you said, maybe we should do a direct experiment of the folding, thermal denaturation experiment. Don't you guys have some fancy way to do this in the QRT machine?
Yep, we have a thermal. It's like a thermal assay.
Peter, I mean, if you want to do it, it's cool. Whoever wants to do it, it's okay with me. But I bet Peter would be interested to do it.
Oh, yes, I would.
Yeah.
Yeah,
So
if we
okay.
get the protein pure, it's actually like a very simple assay, and we get a readout in like 30, 40 minutes. Like we have
Oh, why don't you
screens
guys
for
work
this.
together? Why don't you guys work together to see if you can get that done? Because I think that would be a great way to directly test this hypothesis. And even if all these things I'm mentioning, this may end up in future papers. So don't, we may put the computation you've developed here into this current paper, and then we'll go forward maybe with future ones. So just realize if I said, oh,
Yeah.
crystallize doesn't mean tomorrow, but these are the things that we should do. And then Robert, from your side, basically, if you can work with Peter and you can develop like a methods paragraph and a results paragraph and some figures that we can put together in the paper, then we're really off to the races. I think Peter has a pretty good draft started. I haven't gotten to it yet because I'm just started on Jeremy's today, but a different paper, but I will get to it soon. So anyway, start thinking about the paper. And I think some of the things, your hypothesis I thought was really good, because before you mentioned it, I wrote this down. This would be something interesting for the discussion. And just to remind what I'm talking about here, you said these simulations that we're doing now, they could really give us an indication if we can use this approach to design new reporters that are sensitive to mistranslation, or maybe in the future that are sensitive to other things.
Yeah.
So I think it's really exciting, and that makes for a really nice discussion sort of segment of this a paper relating to the computation. So anyway, those were my sort of few thoughts.
Yeah. Yeah. Well, like you said, like a good readout. Like if we were thinking in the future, if we would be using this computational pipeline to kind of test other mutants, it would be kind of a pain to have to go in and manually check this whole hydrogen bond network local environment to see if it's being disrupted. What I thought is maybe a better readout would be to look, like you said, at just this precursor and look at its what's called conformation. So that's exactly what we did here. And here's just like every single frame you're seeing is one Rosetta model. And in yellow, you see the mature chromophore. And what I decided to do is I made a little histogram here of, turn this into numbers, as you see a RMSD of just the chromophore compared to, sorry, the precursor compared to the chromophore. So in green, we have wild type. In blue, we have the A145G, which has an pretty much identical conformation distribution. And if we look at the S146P, it's completely different. So I think this could be a readout of whether or not chromophore is, disrupted or not. And I don't show the data here, but I also tested this with 146F, and we see also a completely different distribution. It looks nothing like the S146P, but it also looks nothing like the wild type. So it's, I think, a good readout to be.
That is so cool. Sorry to interrupt you. I'm just going to, one last comment. Jeremy, have him also start thinking about the D129P and maybe some of the other ones that we've dreamed up. This is really promising. Sorry, I have to go.
Yeah, no worries.
I'll be in touch, and you guys keep going on this, and we'll get a paper out soon. This is great.
Yeah, thanks, Pat.
Yep.
Right. So I think this is a good readout here because, sorry, I'll go back, but proper precursor positioning, it's like a multi-step process of chromophore maturation, and proper positioning of that initial precursor is crucial for proper maturation, right? And so if you have the precursor that's not positioned properly, you probably won't have the same maturation kinetics as you would for wild type. And so I'm proposing that this is the reason why we're getting a decrease in fluorescence, is because the chromophore just probably isn't maturing properly. And I'm like really happy that this distribution here, like it worked as well as it did. I didn't think that we'd get such a clean readout, but it does seem like a very simple readout that we could just see say functional or non-functional, right? And if we look at,
Awesome.
sorry.
I was just saying that's awesome, first of all. It looks like it's leaning in one direction. Is that, like, on which side of the tyrosine there is the 146?
Yeah, so actually I'm showing that right here.
Oh, you are, sorry, my bad.
Yeah, no worries. Right, so here we have the wild type, and like all 900 structures are kind of overlaid here. It's a little bit harder to see the distribution with all of them overlaid, but here's the serine 146 and here's the proline 146. And then just in the wild type, you can see there's a distribution between like different conformations of the serine 146. But when we add a proline in there, every single one of the 960 models, the proline is facing downwards and is protruding into the kind of chemical space of that tyrosine. So that's definitely why we're getting a change in chromophore conformation, right? So just for reference, that there's the mature mCherry overlaid, right? So you can see that there's significantly more protrusion into the chromophore space, and that's probably why we're getting dysfunction. Right, so yeah, so we're going to conclude that it's that the precursor is not able to mature into the chromophore, and that's why the mutant's non-functional. And another conclusion here is that the distribution of chromophore RMSD seems to be a good indicator of mCherry function in silico. And so this Rosetta-based computational pipeline could be used to discover new fluorescent mistranslation reporters, like fully in silico. Right, so we've got this first KLC step to produce the precursor structure, and then using the precursor structure, we relax the entire backbone, from which we calculate an RMS density plot. Is what I'll call it, as a readout for function. And another lesson learned while I was doing this project is, well, we know structure dictates function, but really it's changes in structure which drive function. And as structural biologists, like, we kind of intuitively know this, but I don't think we really incorporate this enough into structural analysis when we're looking at proteins. And what I'm proposing is, when using a protein structure to probe function, we should also consider dynamics, distribution profiles. So for example, like an RMSD versus energy distribution, but there's lots of other analytics that you can use to take a look at how your different protein models are clustering, and as well as different density profiles. Rather than looking at just a single structure, we take a look at more global metrics such as this. And I like to call this the triple D of structural biology. And I've been trying to incorporate this into, like, whenever I do any sort of structure function projects.
You put Guy Fieri in all of your projects?
Yeah, I call him up before everyone.
Well, that's so awesome, man. This is great.
Yeah, so yeah, hopefully some of that is usable in the paper. If not, maybe we can write a methods paper together, because yes, this could be used to study, like, to kind of generate new mistranslation reporters, but I think just in general, like, you can look at it should be able to apply to, like, any mCherry mutant or even other fluorescent proteins.
Yeah, I feel like it's a great, like, application just for playing around in structure modeling, because the readout of if the protein, like, functions is so simple. It's just fluorescence. It's not like you have to do some enzyme assay to see if your mutant still does stuff.
Yeah, exactly. So we can like validate it pretty easily.
Yeah, that's sweet. One thing I think I've mentioned this before. I don't know if you put more thought into it, but it'd be really cool if we could try to figure out a is it glycine to alanine reporter. I don't know if you tried that. It's a tough challenge, but that's like one of the most common potential mistranslating tRNAs. We get either glycine tRNAs that we think will be mischarged with alanine, or alanine tRNAs with a glycine anticodon. Those have showed up in a bunch of sequencing. So I don't know if you could like apply what you learned here to try like every alanine in the structure.
So I've done that, except I actually mutated, every single alanine to a glycine. But I did that back when our computational pipeline was, well, wrong, didn't really work. I haven't applied it yet to this one, so it'd be pretty simple to do so. I'm gonna write like some Python scripts to automate the whole process, just so you put in a mutant and it spits out that density plot. So yeah, if you have any ideas on like,
I think
what you'd like to apply to this,
and I'll send you this paper. One of the things they did was they did just like MS of basal mistranslation rates in cancer cells. They look at mistranslating tRNAs later, but like their figure 1, they just established like the base rates. I just pulled it up now. So the most common is serine alanine, and then gly-ala, and then serine threonine. So like serine alanine, probably easy to devise a reporter for, but gly-ala and serine threonine are both pretty tricky to just kind of guess.
Yeah, exactly. You'd have
so
to.
I think
it would have to work by some like crazy backbone propagating mutant. I would have to do something that we can't really think of.
Yeah.
But no,
so
it'd be easy to just put through the pipeline. Yeah, you're right.
Yeah, that'd be sweet. Yeah, because like for this one, I just designed it by looking at the structure, and like it's pretty obvious that that serine was hydrogen bonding. So, but if I don't know, you could try putting a threonine there at that 146 position, like S146T maybe.
Okay. Yeah, I've tried it with the phenylalanine, and also the readout was non-functional.
Yeah.
So I can try alanine.
I wonder if threonine might still work fine.
Sorry.
It could be an interesting like, or actually, could it be? Yeah, threonine. Yeah, because the reporter should be basically the opposite of the mistranslation event we're looking for. So if the serine to threonine is a common mistranslation, we want to make that same mutation. Or no, sorry, we want to make the opposite. I'm confusing myself. We would want actually a threonine to serine reporter, sorry. And then an ala to gly reporter. So the mistranslation reverts it to wild type.
Yeah, how about like you just make like a list, even if the list is huge, of like all the mutants you'd like to test. And I'll just take that list and put them all through the pipeline and see what we get.
Yeah, I could start looking at the structure again, or maybe Peter could help me with that.
All right.
Even if it's just like, you want to test all the alanine to glycine mutants, or like all serine threonine.
Yeah, for those we might just want to
test
try them all, because they're going to be pretty subtle.
Yeah.
You might have to just get lucky to find one that actually matters.
Yeah, no, that sounds good. Peter, like, I don't know if you were planning on putting any of this, maybe like a mechanism or something in your paper. If you were, like, which of these figures do you think would fit the best?
That's a good question.
Here, let me share my screen again. Like I think, yeah,
I think
like this
this one.
density plot.
Yeah,
And then
this one really gets the message across.
and if we had it like multi-panel or something, we can maybe put like this just to kind of explain
highlight.
structurally what's going on.
Yeah.
So I was thinking, yeah, we had like wild type S146P, and whether or not you wanted to have this this control, I could, if you want, I could even put in S146F as another control.
You
Or we
can, but
leave it at
I think
this.
this is good as it is right now.
Okay.
But yeah, if I can make a panel between these two figures, that will really.
I'll be able to use the S146F on one of my future papers if I do more work on it. So I've been studying a fetus serum as translator, but I just tried it in human cells, and the mistranslation rate is like crazy low. The human cells really resist the mistranslation, so we might want to try that in like coli or something first. So we have some real data to back it up.
Okay, well, I can send you some of these figures then, Peter, and then if you want me to change them up at all in terms of like the size or the resolution or something.
All right, I'll let you know.
And then if you want, I could like either I can put them into panels and organize them, or I could just give you the raw images and you can do that.
If you know how to put them into panels, you might as well do that right away.
Sure. Okay.
All right. Thank you, Rob.
Yeah, no worries, Peter. Do you have an idea of when you want to submit this? Like,
ideally, the sooner the better. But honestly, it's now in Pat's hand how quickly he wants to get through everything. I'm still aiming for like an end of July, or at least get it to the editors by the end of July.
Okay, sounds good.
I imagine it'll be a little later than that, Peter, because
yeah,
just knowing Pat's
Pat's style
availability. I sent him my paper in like March, I think. But I mean, it's been different because he was on paternity leave. But
yeah, and COVID.
I wouldn't expect him to have it out in 2 weeks. He's going through my paper right now first, so.
All right.
Might be a bit of time, but you can send me a draft too. I'm happy to do around edits.
All right.
A little faster down the road.
Sure. I can send you a draft at some point this week, but if you ever just want to look at my progress, it's in my Dropbox.
Yeah, well, I'd rather just see a draft where it's organized.
Sure.
Because I don't want to be like fishing through random files that I don't really know what they are, right?
All right, fair enough. I can send you my rough draft then soon.
Yeah, it doesn't matter how rough it is. Like, just start getting some words on paper.
All right. Well done. I think this is a very productive meeting.
Yeah,
I know you can't. I know you can't see it, but I'm actually smiling ear to ear. This is really good work, Rob.
Yeah, like it wasn't until like a couple days ago that I decided to do this density thing. Like before, my readout was just looking at the funnel plots, and I was just like, why? I really doubted this. I'm like, are these models even correct? Like, but then yeah, I figured I would just try this and take a look at distribution, and it was all the hardest part was just finding a proper readout. That was like generating the structures was pretty simple. It's the analysis.
Yeah, it's surprising the P didn't change the structure really. I guess I got to start looking at like Ramachandran angles when I'm trying to design things like this. You kind of just assume a proline's gonna screw up the backbone, but
well,
I guess
it did to some extent, but definitely I don't think it was enough to mess up the entire structure.
Yeah, because it's probably like the backbone was probably already close to a proline's preferred angles, right? So I bet it's just if it's like I don't know too much of it. You're the expert here, but if the natural state is at like Ramakrishnan angles that proline can't handle at all, then it would probably be a much bigger perturbation, right?
Yeah, I mean, yeah, if you just like look at how this angle is here, it's very close to what proline wanted.
Yeah. I guess we don't really need more proline reporters, but I should have checked the phi and psi angles of it.
Well, I mean, it's not, I mean, you can't take everything into consideration.
Yeah.
That's why I like looking at these kind of global statistics, or like now whenever I generate Rosetta models, I take a look at this, like the the mean difference between like phi psi angles of all the structures. And yeah, I just like made a little Python script that just colors all of them. So it's just easier to look at.
Oh, you actually wrote the coloring script yourself?
Yeah. There's nothing in Python
sweet.
or in PyMol that does that.
Oh, that's really cool. So that's like another novel thing. Yeah, it might
I guess
almost
I was
be
thinking of putting it on GitHub and like.
Yeah, I wonder if it would almost be worth you would like just also writing your own kind of sister paper to talk about all the, if you wrote scripts and stuff. Like a methods paper, because this is really cool how you can make these figures.
Yeah, maybe. I mean, I'm trying not to spread myself too thin right now. But,
You're like, I don't want more stuff.
But all right, Peter, I'll send you these figures, or I'll try and make like a nice, I don't know, 4-panel figure.
All right.
Did you want maybe this functional mutant in there, or should I just omit it and look at just wild type and S146B?
Keep the functional mutant. I think it strengthens the point even more.
Yeah. Okay. All right. I'll send you what I got, and then I'll send you a little methods paragraph. I didn't send one before because I didn't really have a readout until like a few days ago. So now.
All right. Fair enough. And it seems it won't be as quick as I'd imagined. So whenever you got that methods paragraph, just send it my way.
Sure.
Real quick, Peter, have you tried the 145G again? Have you done anything with that?
No, I haven't touched 145G in a while.
We need to make sure to get, because that was Rashmi that did that. So we need to put Rashmi on the paper because she cloned the reporters.
Gotcha.
And get her data for the 145G fluorescence. And if it's like needs to be repeated, you might want to just transform it again and quantitate it if we're going to include this.
Gotcha.
But it should be easy. You just need to transform the plasmid and check the fluorescence.
All right. Would the plasmids be in Rashmi's box still, or would that be in someone else's box?
Yeah, I think so. I'll help you find them. I think she made strain stocks that I have. So just let me know if you need help finding anything.
Sure thing.
Yeah.
All right. So I think that's it for now.
All right.
Sweet.
Thanks, guys. See you, Jeremy.
Thanks,
See you, Peter.
Yeah.